Hello students and welcome back. This is module 1.2.5. This is actually a second lecture that is given over the same set of slides, and that's broken naturally into the first half, which we've just covered, which is Introduction to Chemistry and Inorganic Chemistry, and the section that we're just beginning on, which is Organic Chemistry. And really what we do there is, as you see on the screen of your computer, biochemistry or biological macromolecules. Macromolecules are what organisms are made out of, macro being large. Biological macromolecules contain carbon. Most are built on a polymer system. A polymer is a long entity made of many subunits. These subunits are monomers. As I say, the vast majority of biological macromolecules are longer, complex molecules made out of repeating units, monomers. There are four major categories of biological macromolecules, and we can see these on this table. If we go across the row at the top, it says the type, the monomer out of which that type is made, some of the major functions and examples. And we'll go through this table very quickly together, and then the balance of the lecture will be describing these categories of biological macromolecules. Carbohydrates are the first type. They follow the polymer model very well. The monomers are simple sugars, for example, glucose. They have two major functions, a structural function and an energy storage function. And there are four examples that we are going to discuss in detail in lecture. That won't be true for the other categories, but we will learn these four examples of carbohydrates, as I say, in fair detail. The second major category is lipids. And of the four categories of biological macromolecules, lipids are the one that do not fit the polymer-monomer model very well. Even though we say that fatty acids are the monomers, that's not true of all lipids. And as we describe these, you'll see the exceptions. The major functions of lipids are structural and as energy storage. Many people think of fat, which is of course one of the major lipids, as being a source of energy storage. The examples here: triglycerides, phospholipids, steroids, and waxes. We'll learn somewhat about the first three and really skip over waxes pretty much. The third category of biological macromolecules are proteins. These fit the polymer model very well. Proteins are long chains made out of repeating subunits or monomers. The monomers are called amino acids, and there are 20 different amino acids that make up all the proteins in the human body. Notice when we look at the functions for proteins that they share structural and energy storage, but then they have two additional functions. Almost, but not quite, unique to proteins is the function of enzymes, or an enzymatic function, and we'll talk about that. And proteins are also, I should say, contractile. Think of muscle fibers. And here are four examples of proteins. And although we'll talk about hemoglobin and we won't talk necessarily about these others so very much. Lactase is an enzyme, you can tell by the suffix ase, and keratin and collagen are important animal structural proteins. The fourth and final category of biological macromolecules are nucleic acids. And like proteins, nucleic acids fit the polymer model very well. They're long chains, long linear chains made out of repeated subunits or repeated monomers. And in this case, the monomers are nucleotides. And there are 5 different nucleotides, although we're not going to be learning about them at this time. The major functions of nucleic acids are information storage, which is really quite unique for a molecule, a chemical, to store information. They also have a function in energy storage, and 3 major types, DNA, RNA, and ATP. And we are going to defer our detailed discussion of nucleic acids until they have a dedicated unit, which will be toward the middle of the semester. before we go on and discuss any of these categories of biological macromolecules, we want to consider how these molecules are put together and taken apart, how they are assembled and disassembled. So there are two reactions that we're going to learn to help us do that: the dehydration synthesis reaction and the hydrolysis reaction. And again, this is an introductory course, but we want you to have a major overview type understanding of these reactions without getting into detailed atoms and such. So here is the dehydration synthesis reaction. And again, when we're looking at names that use this type of language, an etymological approach is very good. Dehydration is removing water and synthesis is building. And so the dehydration synthesis reaction is removing water to build. And notice it is a synthesis or it builds. That means you're forming chemical bonds. That requires an input of energy. And so this is how you, of course, would build polymers. So this is the process by which monomers are bonded together by the removal of a water molecule to build a polymer. And you, of course, you make a long polymer chain by hooking together two monomers at a time. As I've already said, this process requires an energy input and it forms high energy bonds. It's not important. Fructose and glucose make sucrose. Fructose and glucose are two monomers, and this is a disaccharide or a double sugar. And this is just showing, that's just given as an example. Don't even worry about that. But really pay attention to this little diagram down here. On the left, it shows what could be a very long chain. In this case, it's a chain of only two. And notice it ends with a hydrogen. And we're going to add a monomer that has a hydroxyl group. And when the two go together, there are enzymes which remove the hydrogen and the hydroxide groups and form a molecule of water. And as those groups are removed, a chemical bond is formed. And this is where the energy is stored. And so by removing water and forming that bond, that is what a dehydration synthesis is. Now we can look at the other side of the coin, and that is how we take one of these long polymers and break off the constituent monomer, the single unit. If we look at the name of a hydrolysis reaction, hydro is water and lyse, L-Y-S-E, is splitting. Hydrolysis is splitting with water. And so we're breaking chemical bonds. And so this is a process by which polymers are broken down into monomers by adding water molecules, or also breaking high energy bonds to release energy. Once again, if you just pay attention to this diagram here, in this case we have a chain of three, but it could be a chain of 1,000. But it's only on the end where the hydrolysis reaction occurs. And so there are enzymes which will help to bring a molecule of water in and split that molecule of water, breaking the bond at the end of the polymer and putting the hydrogen group on one side and the hydroxide group on the other, and thereby breaking the bond, as I said, liberating energy and forming a separate polymer, you can see. I'm sorry, I misspoke. That would be a separate monomer and leaving the polymer intact over there. We're going to conclude this module just by looking ahead. This is what we're going to be discussing next time, the 4 categories of biological macromolecules. So we will get to that later. Thank you very much.